Hello, everyone, and welcome to today's webinar, Tips and Tricks for Automating Viscous Liquid Handling. I am Sydney McNeil of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Opentrons. To learn more, visit opentrons.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of our presentation. You may also submit any technical issues here as well, if you have any trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Anurag Kanase, Process Development Scientist, Opentrons. Anurag, you may now begin your presentation.
Thank you, Sydney, very much. Thank you, everyone, for joining today's seminar. And in this seminar, I would like to introduce you to understand the different properties of viscous liquids and how we can use automated liquid handling and optimize these parameters. So after handling the viscous liquids, the parameters, you'll be able to accurately handle viscous liquids using the OT2, able to reduce the reagent wastage and avoid cross-contamination across your deck, and finally improve the reliability of the protocols running the viscous liquids. Before we begin, let me first introduce you two important concepts concerning the viscous liquids. The first one you see on the left-hand side is a picture containing a tip with liquid, which is glycerol, dangling around the tip. So on the top region, you see the liquid is present inside the tip, and somewhere around the meniscus is broken of the liquid, and at the bottom, the liquid is dripping around the tip. So these forces where the liquid is adhering to the labware is known as adhesion. And on the right-hand side is glycerol being poured in the cap of the bottom cap. So the forces which are holding together the glycerol are the intermolecular forces, and the bonds which hold together glycerol is known as cohesive forces. We'll be using these concepts further in the presentation. As a basic guideline, let's take an example of how liquid flows through the tip. For simplicity, we are considering the tip as a pipe. And on the left-hand side, we see a pipe, and the liquid is flowing through the pipe. So when you're pressing the plunger of your pipette, what happens is the liquid starts flowing, and there are a lot of friction forces at the boundary of the tip or the wall of the tip. And these forces causes the liquid on the boundary region to flow slowly. So these forces, which are frictional forces, are known as adhesive forces in case of this particular use case. And the liquid starts flowing in the center. The liquid starts flowing at a faster pace. So these forces which bind the liquid to move further are known as cohesive forces. So now if we start looking at the flow profile of the liquid, which is on the right-hand side, the maximum resistance is at the boundary over here, at the boundary of the tip. And as we go towards the center where the cohesive forces are much larger, we see the flow pattern is more like in a parabolic shape. And at the center, there is the least resistance due to the cohesive forces, and on the wall, the maximum resistance due to the adhesive forces. Now, the strategy to handle viscous liquid is to minimize or get this red arrow as much as closer as possible towards the beginning of the meniscus, which means that we have to align the ratio between the adhesive forces and the cohesive forces for having an effective flow through the pipette tip. Now, based on the relative ratio of these adhesive and the cohesive forces, which dominate inside the tip for a given liquid, we are going to characterize the viscous liquid classes. So we have characterized viscous liquid classes into 4 different categories. The first one is glycated liquids. The glycated liquids are essentially glycerol-like liquids, or more a vague term would be sugary liquids. So these example includes honey and honey glycerol or PEGs. These kind of liquids have very high adhesion and high cohesive forces. The second category is volatile viscous liquids. An example would be beads, hydrophobic coatings, or hand sanitizer. So these liquids have very high adhesion to the tip and neutral cohesion to the liquid itself. The third category is surfactant viscous liquids. These viscous liquids are essentially soapy liquids or detergents, but they are viscous in nature. For example, Tween Buffer or Triton X or liquid hand washes. They show very high adhesion to the tip wall, but very low cohesion to itself. So that's the particular property of the surfactant itself. And the fourth category is oils. An oils example of oils may include mineral oil, engine oil, or cooking oils, and they show very high adhesion to the tip wall, also very high adhesion to the liquid. Let's take an example of what exactly happens when a liquid is aspirated, a viscous liquid is aspirated. When any robot or even the manual pipetter ship, they are tuned for best performance using water. So if you're going to use the same parameters to handle viscous liquids, this is the case which is going to happen. This is a case of aspiration of viscous liquid. The robot moves the tip pretty fast, and then it tries to aspirate the liquid, and it doesn't allow sufficient time for the liquid to get aspirated, and we can see the liquid being incompletely aspirated. The droplets are formed around the tip, and there is spilling of a reagent. Now, let's look at what happens if you're going to dispense the same liquid. So when we insert the tip, we can see that a lot of liquid was wasted around the walls, and the tip is not completely dispensed. There was wastage of reagent and overflowing reagent, and such volumes are non-reproducible. So the idea is to actually optimize these parameters based on the flow rate of the liquid and how the tip is being aspirated, dispensed, and if there is any dispense time and the withdrawal of the tip. So let's take a look at how we can optimize any viscous liquid for smoother liquid handling. The idea is to first of all have an aspiration flow rate to the 1/10 of whatever your flow rate was defined for water. So in Opentrons case, we're using 27 microliter for P1000 as the aspiration for microliter per second for aspiration. We delay for 20 seconds after aspiration, and then we withdraw the tip at 400 millimeters per second. Now, let's try to dispense this liquid at the same speed as what we are dispensing water. So what happens is the meniscus is broken immediately, and the liquid or the plunger has completed its complete displacement. Now we see that some liquid is being dispensed towards the bottom of the meniscus, and the liquid is not completely dispensed out of the tip. So the next step is to reduce the flow rate. Now the idea is to get reduce the flow rate such that the meniscus is gradually decreasing until the mid of the tip. So we are using a dispense flow rate of 1/6 of water, and we're using the same blowout rate. Let's try to dispense at 1/6 of the rate. We saw that earlier the meniscus was broken over here, but now we have broken the meniscus little below. But again, we see the liquid is not completely dispensed out of the tip. And now let's try to optimize it further. Let's try to reduce the flow rate. Now we are going to reduce the flow rate to 1/10 of the water flow rate and add a delay of 10 seconds after the liquid is being dispensed, and then perform a blowout. So the blowout which we are using is the normal blowout. And now we can see the liquid is slowly dispensed compared to previous, and the meniscus is broken much below the tip. Now after dispensing, a normal blowout flow rate was used, and still there is some kind of liquid, which is still present inside the tip. Now this is glycerol 99%. Now, finally, what we would like to do is we are going to dispense at 1/10 of the water rate, which we earlier realized it's a pretty good rate for this particular liquid, add a delay of 10 seconds, but we are also going to decrease the blowout flow rate, which allows the liquid, which is settled at the bottom, to slowly get removed from the tip. Let's take a look over here. Now we are slowly dispensing the liquid, and now there is a delay for 10 seconds, and a slow blowout of 1/55 of that of water allowed us to have a pretty good tip. Let's take this optimized strategy for viscous liquid handling. So if you're going to aspirate the liquid, move to the top of the lather at the default speed. Then move to the bottom of the lather at a slower speed, which is 10 millimeters per second. And then aspirate at a slower flow rate and add a delay so that the liquid, which is still being aspirated due to the negative pressure which is being developed inside the tip and the plunger head, is allowing the liquid to get settled towards the top of the tip or to the maximum volume of the tip. And finally, withdraw the tip slowly from the lather at 1 to 5 millimeter per second. Further, to dispense viscous liquid, we're going to move towards the top of the lather and then move to the bottom of the lather at 10 millimeters per second. This is to reduce any kind of spilling of the liquid, which is already present inside the lather, and then dispense at a slower flow rate. Further, we are going to add a delay or some kind of delay, which will help the liquid to dispense completely and also settle down to the bottom of the tip. Now, once there is residual volume present at the bottom of the tip, we are going to remove this using a slower blowout. And after using a slower blowout, we are going to withdraw the tip from the liquid at a slower flow rate, which is at a slower withdrawal rate, which is 1 to 5 millimeter per second. Let's take an example in the case of glycerol liquids, how we are going to handle the glycerol liquids now. So the common properties of the glycerol liquids are high cohesive forces and high adhesion to the tip. So there is a pretty good synchronization between the meniscus which is formed and the flow rate. So all you have to do for the glycerol liquids is to tune them such that the meniscus is not broken until the liquid has reached 1/3 of the tip position, or where the tip actually starts narrowing down. So this liquid, if there is any kind of to the tip, it doesn't really remove off because there is no gravity being acting on it. And now let's take an example of how to optimize for glycerol 99%. So the first step of aspirating the liquid, what I'm going to do is I'm going to aspirate it at 1/10 of the normal flow rate. And after aspiration, I'm going to provide a delay of 20 seconds so that the liquid is completely aspirated inside the tip. And then I'm going to withdraw the tip at 1 millimeter per second. The viscosity of glycerol 99% roughly over here is 890 centipoise. So here I go. Now we immerse the tip and we aspirate at a slower flow rate. The video speed is like 8x. After the delay, we are slowly removing the tip from the liquid. Therefore, what we can see is the pipette is able to highly accurately aspirate the liquid. It's a very clean pipetting. There is no spilling around and no droplets are formed around the tip. The only drawback over here is the longer protocol time. Now, let's take an example how we can do the dispense cycle after the liquid is being aspirated inside the tip. So for dispensing it, the dispense flow rate is 1/10 of the default dispense speed, flow rate, and the dispense delay is 20 seconds. So after dispensing, we are going to wait for 20 seconds, and then the blowout is being performed at 1/55 of the default water flow rate, and the tip withdrawal speed is 1 millimeter per second. So all these actions can be performed using the Python API for Opentrons. Here we go. We are dispensing it at 1/10. After the dispense is complete, we are waiting for 20 seconds. Now we are withdrawing the tip a little bit above the surface and blowout was performed. The withdrawal was done at 1 millimeter per second, and you can see the tip is pretty clean. And there is no spilling around, and minimal droplets are being sticking to the tip. So the idea to handle the viscous liquid is lower your flow rate, reduce the blowout rate, and add a delay after your aspiration or dispense cycle. The most critical tip is to withdraw the tip at slowest speed if the viscosity of your glycerol liquid is really high. For instance, in case of if you're going to aspirate honey kind of liquid. Now, let's look at what happens with the volatile viscous liquids. So the volatile viscous liquids are essentially solvents which are mixed with some kind of bead solution or maybe glycerol. For instance, in case of hydrophobic coatings, or maybe beads, or hand sanitizer would be the best, easiest example. So volatile viscous liquids have very high adhesion to the tip. So they keep on sticking to the tip, but they have low cohesion to itself. So you can see the surface property of the volatile liquids is really poor. They don't stick to each other. And the volatility may really vary depending on the volatile liquid or volatile solvent that's being used. The critical factor over here is to use air gap because if you're going to dispense it, there is a jet going to form and the liquid is still going to stick to the wall. Now let's take an example of how we are going to handle the hand sanitizer, which is having a viscosity of roughly around 200 centipoise. So we're going to use similar parameters which we use for glycerol, and we're going to aspirate it at 1/10 of the default flow rate, then wait for 20 seconds after aspiration, and then withdraw it at 1 millimeter per second. The good thing about aspirating viscous solvents is they are a little easy to aspirate. Now we are going to withdraw it. And as you can see, there was no droplet forming around the tip, but we definitely need an air gap to avoid any kind of spilling of liquid if there is pressure built inside the upper cavity of the tip and the plunger, between the top surface of the liquid and the plunger. So we see a clean tip and there's no spilling and no droplets are formed. Now let's take a look at how we can dispense it. Now, an important factor over here is, as we discussed earlier, these liquids or volatile liquids have very high adhesive forces, which means that the liquid is sticking to the laver or has very high affinity to the laver. So what we are going to use a method called as touch dispense, in which what you do is you move the tip to the wall of the laver and then dispense at a slower flow rate, which is 1/10 of the flow rate, default flow rate, add a dispense delay of 20 seconds, and then we are going to perform a slower blowout. Let's take a look at this example. Now, over here, we are doing a touch dispense where we are sticking the tip to the wall of the laver, and we are dispensing. Now we are delaying it for 20 seconds, and we are going to perform the blowout. We can still see that there is some residual volume which is sticking to the tip. The problem with such liquids is either you can dispense it in multiple steps, like you dispense half of the liquid in the tip and then allow the liquid which has been sticking to the wall to gradually settle down to the orifice of towards the orifice of the tip. This helps in reducing your protocol runtime. But the trade-off is you'll have a longer protocol runtime, and there will always be some residual volume left. But considering depending on the application of your protocol, you can consider what the residual volume is going to be. Now let's take a look at the surfactant viscous liquids. The basic property of surfactant viscous liquids are they are surfactants or detergents, very poor surface tension, and they don't have any adhesion to itself or very poor adhesion to itself, but they have very high adhesion to the laver which you're using. They behave a little similar to the volatile liquids which we saw earlier, and the examples would be tween buffer or Triton X, which are commonly used in our protocols. And the world famous example would be liquid hand soap. So if you're going to dispense this liquid, the common problems which you might be having is there is excess retention of liquid after the viscous liquid is being dispensed. So you might need to dispense in multi-steps where you're allowing the surfactant to completely come to the bottom of the orifice of the tip. Let's take an example of how we are doing this for Triton X. So the viscosity of Triton X is around 230 centipoise. We're using lower concentration of dye just to avoid any kind of bubbling problem. And these liquids have very easy affinity for creating bubbles around the liquid, which might get trapped inside your tip. So it's always a good recommendation that when you're starting your protocol, try to avoid the bubbles in your lab there itself. Here we're going to use, again, 1/10 of the flow rate, and the aspiration rate is going to be 1/10 of your default flow rate. Your aspiration delay is going to be 20 seconds, and the tip withdrawal speed is going to be 1 millimeter per second. So we're going to slowly aspirate. We can see the liquid is being aspirated properly, and since there it required some liquid which was sticking to the laver, and the tip was being touching to the laver, it caused some liquid not to get aspirated. It's again a property of viscous liquid, but when the basic property of the surfactant handling surfactant liquids is these liquids have very high adhesive forces, which means that they'll keep on sticking to the laver when you're aspirating it. After aspiration, there might be some kind of hollow gap or a hollow cavity which is being created inside the liquid, which may cause a problem like a bubble being introduced or air gap being introduced after the liquid is aspirated. So just to make sure, you're completely going to aspirate and wait until the liquid has settled down, and then only withdraw the tip. So in this liquid, we can see there is clean pipetting, no bubbles being formed, and no spilling around. Also, no droplets are formed, but there is an air gap problem, which depending on the need of your protocol. Now, let's take a look at how we are going to dispense this viscous liquid. Again, coming back to the affinity of the liquid to the laver, we have to dispense at the top, because now when we are dispensing this liquid, it will easily adhere to the top or the tip itself when it is being dispensed out. So it's a good idea to dispense at the top of the wall and then dispense at a slower flow rate, so that the whole liquid, we are still able to maintain the meniscus, and the whole liquid is able to flow cohesively towards the bottom of the tip. Introduce a delay of 20 seconds, and if one dispense is not enough, you can add two dispense of the liquids, or multi-dispense, in which you're going to split the dispense cycle into two parts. For instance, for P1000, you can dispense with 500 microliters first, and then again wait for some time and dispense 500 microliters during the second time. This increases your protocol runtime, but let's take a look how this behaves. We're dispensing at the top of the surface, but you can see the liquid is still taking some time to fall towards the bottom of the tip. Now we are going to perform a blowout, and over here there is still some liquid which is being retained inside the tip. So if protocol runtime is a critical factor in your protocol, then it's a good way to estimate what will be the excess liquid or residual volume which is being observed inside the tip, and then consider optimizing your protocol based on the residual volume, or based on the performance. If you want to compensate using a time of faster time, then use multi-dispense steps, or just consider there will be some residual volume, yet you can dispense it at a very higher speed. Or you have to, if you want a complete clean dispense, then longer protocol runtime will be a problem. So that's a kind of a trade-off between faster protocol and a slower protocol for surfactant liquids, and like the residual volume versus the runtime. Final liquids class is oils. So the oils are any kind of oils which are being used. For example, it would be CBD oil, mineral oil, or plant oil, which are being pipetted for your protocol purposes. So they have very high adhesion, but they also show very high cohesion to the liquid itself. So they do relatively move to the tip, but the problem is, due to very high adhesive forces, they keep on sticking to the wall of the tip. Slowing the flow rates allow easy aspiration dispense. So generally, it's pretty easy to aspirate and dispense at slower flow rates. But again, similar to surfactants, oils also retain liquid inside after the dispense cycle. So let's take an example of olive oil being dispensed. The viscosity of olive oil used over here is roughly around 90 centipoise. And what we are going to do is aspirate at a slower flow rate, slowly withdraw the tip after allowing a delay of 20 seconds after aspiration. We introduce an slower aspiration flow rate, allow some delay, and then withdraw the tip. Um, in most of the videos, you may have noticed that the liquid is still being aspirated and suddenly the tip starts moving up. The phenomenon which is happening over here is the slow aspiration is taking place inside the tip. The liquid is slowly being aspirated during the delay cycle. So you always continuously see that, even though the robot is not running or the pipette plunger is not moving, but the liquid is still climbing negative pressure and because of the viscosity slowly. And so this aspiration delay time allows you for the liquid to completely climb up the tip and fully attain the desired target volume. And now we are going to slowly withdraw the tip. You can see slowly withdrawing the tip actually reduces a lot of droplet problem, and there is no spilling outside your labware. So it's a very good strategy to remove for viscous liquids or volatile liquids. It slowly remove your tip outside the labware. In the next step, what we're going to do is dispense the same liquid. As I told you, it has a very high affinity to the wall of the tip. And oils also behave similar to surfactants such that they will keep on sticking to the tip inside for a longer period of time. So there will be some residual volume left, but it's less compared to what we saw in case of surfactant. And we are going to dispense at the top just to take advantage of the adhesive forces so that the liquid is still quickly sticking to the labware while it is flowing inside the labware from the tip. So let's take a look. First step, we go and dispense at the top of the labware at a slower speed. This is a real-time video. So this is the amount of time which is required for the liquid to get dispensed properly. I believe at this point that the dispense delay has already started working on it. And so the slower blowout was introduced, and you can see there is very less liquid which can be observed right now. But if gradually allowing some time, the oil which is sticking to the wall of the tip will start settling down to the bottom of the tip. So there will be some residual volume left, and we have to be mindful while optimizing our protocol. And again, it's a pretty longer protocol time. So the viscous liquids, in general, they have a longer run time, but if you are able to wait for that, if you're able to have a trade-off of that time, you're able to achieve really good and accurate results to your target volumes for your desired protocols. These are the examples of gravimetric test results. And this was done with glycerol, different concentration of glycerol as they are representative classes of our viscous liquids, because glycerol 10% has a viscosity of roughly around 2 to 3 centipoise, 90% has a viscosity of roughly around 160 centipoise, and 99% has a viscosity of around 890 centipoise. So these three different classes cover a varied range of viscosity, and the gravimetric testing was performed on these liquids using the P20. So the results are very comparable to our water results, or how P20 can handle water, as well as for P300, the results are very similar to how it can handle water in different liquid cases. For P1000, again, it's a very similar case. The glycerol handling for 99%, 90%, and 10%, it's very similar. The results look pretty similar to water. So we're able to use these kind of techniques to definitely smooth and use a controlled flow rate using the automated liquid handlers to handle viscous liquids, which are not possible if you're dispensing it manually, or if you're using manual processes for your protocols. So, and we are able to achieve accuracy, which we showed in the results part, which is very close to our water CV. And this accuracy is very accuracy for aspiration and dispense for the desired volume. Finally, we're able to increase the reproducibility in the results, reduce the reagent wastage, and do this completely automated without any human interference. I would like to thank our testing team, Carlos and Ujis, for helping during the testing time. and I'm open to your questions. Thank you.
Thank you, Anurag, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, could you elaborate on mixing viscous liquids?
Yeah, that's a very good question. Mixing viscous liquids is a little tricky affair. It really depends on what kind of labware you're using. So first of all, if you're going to use very tall labware, for instance, like 15 ml tube or like 50 ml tube, and depending on the volume which is present inside your labware, it's difficult to mix if the volume inside your 15 ml Falcon tube is to the maximum. So the idea is to go to the bottom of the labware and try to mix it slowly several times. One mix or two mix is not really going to help you mixing it. Start with a viscous liquid and dispense into the labware, and then dispense a less viscous liquid. Mix it, try to optimize your protocol at mixing at slower flow rate, which you used for the higher viscous liquid. And once you are able to achieve, try to go through several mixing steps and try to optimize your protocol based on the two different liquids which you're using. Another way is to, if your protocol is able to support, to increase the temperature of your liquid. For instance, if you're going to work with 99% glycerol, and if you're able to heat the glycerol to higher temperature, for instance, like 50 degrees Celsius, the viscosity of the glycerol drops gradually. I think it drops at 50 degrees Celsius, it drops to like around 1/5 of whatever the current viscosity is. So it's a good idea to increase the temperature if your protocol allows you to increase the temperature and then mix together the viscous liquids.
Perfect, thank you. Our next question is, what do you recommend to reduce bubbles during blowouts, especially in detergents?
Very interesting question. So to reduce the bubbles during the blowout, the most critical step is insert your tip to the top of the labware and dispense it, do something as touch dispense. But also another important thing is make sure your tip doesn't have any kind of air gaps. So if let's say you're going to dispense 100 microliter with P300, then slowly immerse your tip on the top of the liquid, making sure that there is no air gap, and dispense 99 microliter only because that 1 microliter of liquid which is present inside your tip won't introduce any kind of air gap. So that would be a good way to not incorporate any kind of external air or air which is already present in the tip, and just dispense the liquid almost to 99%. So that reduces any kind of bubbling problem, especially for detergents.
Great, thank you. Our next question, could you comment on changing pipetting parameters as the temperature decreases and increases liquid viscosity?
Oh yeah, sure. So if you're going to decrease the temperature of the liquid, for example, if you're going to decrease glycerol, and I've tested this at 14 degrees Celsius with our TempDeck module. So the viscosity actually drops down pretty significantly. I think the viscosity the viscosity increases 4 times whatever it was for at 25 degrees Celsius, for instance, compared to 14 degrees. So the idea is to reduce your flow rate. There are some applications which require you to work at lower temperature, for example, like PCR, qPCR applications. So a good way to handle these kind of situation is to slower the flow rate than what you're using it for your current viscosity. It's always a good idea to look at the viscosity chart and then compare your previously used liquid and reduce the flow rate accordingly. And then withdraw the tip slowly. And if you're going to increase the temperature, then the viscosity is decreasing so that you can take advantage of a faster flow rate, faster aspiration and dispense flow rate. In this case, your protocol runtime is decreasing, but if there is any possibility of damaging your protocol, if there is any kind of sensitive matter, material, then it's a good idea not to increase the temperature and keep working at the prescribed temperature.
Great, thank you. Next question. Could you speak on the touch tip function and how they can be helpful in dispensing viscous liquids?
Sure. So the touch tip function is generally used when the liquid is still sticking to the tip, like the orifice of the tip. So a good way to try to remove these kind of liquid droplets are using touch tips. So essentially what it does is the way we do it manually, like we keep on touching the surfaces of the labware so as to remove any kind of liquid which is being attached to the walls. So the touch tip essentially does the same thing, and it can, like you can perform touch tip at different locations inside the labware. Also, depending on the shape of your well, it is important to consider touch tip function. For example, if you're going to use a V-shaped well and you're going to use touch tip at the bottom, then there is a high possibility that you might drop off the tip. So in viscous liquids, use touch tip only when consider what the height you want to use the touch tip at, and use it only when you observe that the liquid droplets are sticking to the tip. This happens mostly in case of beads where there is some kind of droplets are being sticking out at the tip. I would recommend to use touch dispense instead, where you're moving your tip to the edge of the labware and dispensing completely.
Perfect, thank you. Next question. Can the tips themselves have an effect? Different companies, for example.
That's a very good point. Yes, yes. So the reason tips have the reason why tips have different effects on the way you're dispensing is because first is the profile of the tip. The way liquid handlers are programmed are using the tip profile. So like your plunger displacement can help you in dispensing it at based on your tip. If the tip has a larger bore, the plunger displacement can be faster. But if your tip has a slower bore, then it has a different slower plunger displacement rate. So also the second property of the tip is what kind of material or what kind of plastic is being used. So generally, there is a lot of electrostatic forces which are causing the liquid to stick around the walls of the tip. So if you're using a very good quality of the tip, then it would generally have lower electrostatic forces, which are causing lesser droplets being sticking to the tip. And another factor is how good is the packing of the tip to the pipette. So using an incompatible tip is somewhere where there is a small kind of air being present between your tip head and the plunger and the pipette. So what happens is if there is any kind of leakage, you're not able to aspirate it properly. So definitely the tip has like the tip material or the tip shape or the tip brand has a effect on the liquid handling performance. And it's a good idea to use or try out the tips, try to test out the volumes of the liquids using your gravimetric methods, and then only rely on those tips which you think are going to perform well with your liquid handler.
Great, thank you. Next question. What liquid class would Matrigel and similar hydrogels for 3D cell cultures fall in?
That's a good question. I think so. The Matrigel would be more like I think it would be a combination of three classes, not really a volatile class, but still more like a surfactant class because they are hydrogels, and volatile only because there is some kind of retention inside the tip, and the glycerol class. So I think the good way to handle a 3D cell culture using the hydrogels is first of all making sure that your tip is able to aspirate and dispense them properly. Like there is no blocking being performed. The tip is not blocking these, or the hydrogels, or if they are pretty larger in size, they're not blocking it. So that's the first step to find out. And the second thing is going to be reducing the flow rate for Matrigel and hydrogels. And the third step would be withdrawing it slowly. So I think using the first three classes would be easy. Using the touch dispense will definitely help in removing the hydrogels from the tip.
Great, thank you. It looks like we have one final question. Are the aspirate and dispense settings in your presentation standard for each type of viscous liquid?
That's a good question. It can be tuned. The reason I have used these standard functions, or so we definitely have these functions in our app note. Please have a look at the app note for different kinds of liquids which we have tested. And currently, the presented liquids are more like they have been tested gravimetrically, so we can pretty much assure about these parameters. The reasons like these parameters were pretty much consistent are because we found out like these parameters works best. But if you want to have your different, if you have a pretty different liquid, you can definitely begin with the viscosity for like the viscosity as a parameter, and then choose what are the parameters for such liquids. So for instance, if you're working with PEG, which is not mentioned over here right now in the presentation, but in our app note we have something for PEG. So let's say you are having a different concentration of PEG, maybe like 25%. Then what you're going to do is try to consider or try to search for what is the viscosity of this 25% PEG. Let's say it is going to be 50 centipoise. Then what parameters work well for centipoise? 50 centipoise will definitely help you understanding uh, um, in handling these kind of unknown liquids, and try to and the first strategy which I showed you is to understand how, at what range or at what rate you are able to keep the meniscus constant. This is a good way to begin with. So aspirate your liquid at a slower flow rate, and then dispense at a slower flow rate, and try to keep the meniscus constant until it reaches to where the tip starts changing its face or the shape profile of the tip. Uh, I can take a couple of questions if there are any.
It looks like we did have one more come through. Any particular tips for magnetic beads pipetting?
Sure, yeah. So uh, again, the magnetic beads are different, based on the solvent they are present in. It could be like you might be having some kind of glycerol present, which are pretty easy to handle using the glycerol kind of parameters which we stated. But there are other beads which are placed in some water buffer or like ethanol or some kind of detergent, if any. So for ethanol, it's a very critical thing to aspirate the beads at a slower speed and make sure they are mixed properly or homogeneously, and then aspirate it at slower speed, and then use an air gap function because the ethanol is going to drop. So use some volume of air gap, 5 microliters per P300 works well, and then move the tip to the desired position.
Great, thank you. One more question. Where can I find the app note you mentioned? Will it be included in the follow-up email?
Sure, yeah, we can send out the app note in the follow-up email, but the good idea would be to look for the app note at in our app note section on Opentrons website. So you'll be able to find out the app note for viscous liquid handling on the Opentrons website. Keep an eye on the documents section of the Opentrons website.
Perfect. It looks like that is all of the questions we have for today. Anurag, do you have any final comments for our audience?
Thank you very much for attending today's talk, and let me know if there are any kind of questions. If you feel there is something that you need to be covered, or you need some assistance, you can definitely reach out to our AppSense team and reach out to me in case of any technical concerns. If you want your protocols to be developed for handling specific liquids, our AppEng team is and these protocols can be handled pretty well only using Python API. The protocol developer has some limitations at the moment. We are trying on improving the performance of the protocol developer. So the AppSense team will definitely be able to help you using the Python API. And please feel free to reach out to Melissa for any product inquiries. If you have any demo requests of the OD2s, and if you require any particular application, please let Melissa know. She'll be of very great help to you. And thank you, everyone.
Thank you again, Anurag, for your time today. We would also like to thank LabRoots and our sponsor, Opentrons, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.